Cancer is a serious disease that caused the death of 85,000 Canadians in 2021 alone. In over 50% of these cancer cases, a mutation of the p53 gene was identified. The p53 gene produces a protein that detects DNA damage and either repairs the DNA or causes the cell to undergo controlled cellular death, also known as apoptosis. Many treatments have been investigated to determine their effects on the cancerous cells and the regulation of their p53 gene. Resveratrol, being one of those studied compounds, is a polyphenol found in the skins and seeds of grapes and other berries. Some of the highest concentrations of this compound are found in wines. In this research, MD-MBA 231 breast cancer cells were treated with resveratrol to determine the effect it has on cell viability and p53 gene expression. To investigate these questions, three objectives were examined. First, SPE was used to extract resveratrol from the wine before it was quantified using UPLC. Second, breast cancer cells were grown and treated with resveratrol ranging from 1 to 100 micromolar concentrations. After 24 hours, cell viability was determined with an MTT assay. Finally, relative p53 gene expression was compared between the vehicle control of 1% DMSO and the 100 micromolar resveratrol treatment. Resveratrol concentrations in wine samples can be seen in Figure 1. Red and white wine contained 2.18 and 0.231 milligrams per liter of resveratrol respectively. The homemade red wine sample contained no resveratrol. The variability of concentrations is due to different production methods of the wine. Red wine fermentation occurs with the grape skin on, while the skin is removed during white wine production. With most of resveratrol located in the grape skin, it has become clear why the red wine concentration contained higher concentrations of the polyphenol. The homemade red wine, which was produced from a kit, did not include any grape skins, which led to no resveratrol being identified in the wine sample. Cell viability analysis can be viewed in Figure 2. A significant decrease in cell viability was determined in the 50 and 100 micromolar treatments when compared with the vehicle control group. Figure 3, seen on the bottom, displays the relative p53 gene expression comparing the vehicle control to the 100 micromolar treatment. Although a trend toward increased expression was observed, no statistically significant difference was detected between the two groups. The treatment of breast cancer cells with resveratrol led to cell death in concentrations above 50 micromolar, but did not lead to significant difference in p53 gene expression. The quantity of resveratrol found in the red wine, equating to approximately a 10 micromolar concentration, was not enough to evoke these cellular responses. This suggests that resveratrol levels found in wine are approximately 5 times lower than the treatment level required to provide a decrease in cellular viability in breast cancer cells. Determining whether the same decrease in cell viability is observed in healthy cells is the next step in establishing this treatment feasibility. If resveratrol causes the same decrease in cell viability in healthy tissue, the issue with these treatments then becomes the need for direct targeting of cancerous cells with the polyphenol. If the treatment can decrease cell viability on cancerous cells alone, then more research on delivering concentrated forms of resveratrol will be necessary to determine its viability as a cancer treatment. I'd finally like to thank Dr. Jessica Livingston, Dr. Mike Gibson, Marla McKenzie, and Crystal McFadden for all their help in this research project. Thank you.